calling me Heritage, and this is group 3. We'll be dealing on the enzyme hydrolase. Hydrolase is a class of hydrolytic enzyme that commonly perform a biochemical catalyst that use water to break down a chemical bond, which results in the breaking down of larger molecules into smaller molecules. In order for us to understand this enzyme hydrolase very well, we'll look at the examples of the enzyme hydrolase, the characteristics and the properties, the classification of this enzyme, the importance of these enzymes to the human body, and also we'll be looking at the reactions of this enzyme. Come with me as we take you on this journey.
The enzyme hydrolysis. Some of its examples are exteresis, which include lipases, phosphatases, nucleosidases, and glycosidases.
Okay, having looked at the definition of hydrolysis and the example of hydrolysis, now let's look at the characteristics or properties of hydrolysis. Some of the unique characteristics of hydrolysis include 1. Hydrolysis are soluble in water. 2. Hydrolysis have broad substrate specificity. 3. Hydrolysis often have high stereoselectivity, even towards unnatural substrates. Hydrolysis undergoes hydrolysis, which involves the use of water to break chemical bonds in diverse substrates of different sizes and complexity, such as carbohydrates, protein, lipids, and nucleic acid. Hydrolysis have choir cofactors. They tolerate the use of water-miscible solvents such as dimethyl sulfoxide and dimethylformite. Also, hydrolysis are generally extracellular enzymes so that they can be purified easily. Lastly, hydrolysis have degradative properties and have quite characteristic activation energies. They also have substrate-free energies and enzyme efficiency.
Let's take a look at classification of hydrolysis. Hydrolyses are classified based on E.C.N number, that is enzyme commission number. Hydrolysis can also be further classified into several subclasses based on the bonds they act upon. Number 1, EC3.1. These enzymes act on esters groups, which are called esteres. Some common esteres include nucleosidases, phosphatases, and lipases. EC3.2. They act mainly on sugars such as DNA glycosylases and glycoside hydrolases. EC3.3. This enzyme acts on ether bonds, destroying ethers. EC3.4. They act on peptide bonds such as peptidases and proteases. EC3.5. They act on carbon-nitrogen bonds other than peptide bonds. Example, amidase and urate.
Hydrolysis, standing from enzyme commission number 3.6, that is EC3.6, acting on acid and hydrate. Example of these enzymes are inorganic diphosphatase and fat diphosphatase. EC3.7, acting on carbon-carbon bonds. Example of those enzymes are oxaloacetate. EC3.8, acting on halide bonds. Example of those enzymes are 4-chlorobenzoate. EC3.9, acting on phosphorus nitrogen bond. Example of those enzymes are phosphoamidase. EC3.10, acting on sulfur nitrogen bond. Example of those enzymes are cyclimate sulfur hydrolase.
Looking at classification of enzymes according to their enzyme commission number, and we've considered the enzyme commission numbers ranging from 3.1 down to 3.10. Now I would like to take over from where my colleague stopped, which is enzyme commission number 3.11. Now, one of the core enzyme commission number 3.11 has to do with the enzyme that act on carbon phosphorus bond. Now, example of such enzymes which act on carbon phosphorus bond has to do with what the phosphonoacetaldehyde hydrolase. Now, another example of such enzyme is the phosphonopyruvate hydrolase. Now, the next one, which is the enzyme commission number 3.12, which act on sulfur-sulfur bond. Example of such enzymes which act on sulfur-sulfur bond has to do with the tritonate enzymes. or hydraulics. Now the last one, which is the enzyme commission number 3.13, which act on carbon sulfur bond. Now example of such enzymes that normally act on carbon sulfur bond include the carbon disulfide hydraulics, and the next one, which is what the carbony sulfur hydraulics.
Hydrolysis. Biologically, hydrolysis are of crucial importance for the body since they digest large molecules into fragments for synthesis, excrete waste materials, provide carbon sources for the production of energy, during which many biopolymers are converted to monomers. Now, where are these enzymes found in the body? Hydrolyses are found in the FAH, the focal acral hyperkeratosis gene, which provides instructions for making an enzyme called fumaryl acetoacetate hydrolase. Now, this enzyme is abundant in the liver and kidneys and smaller amounts throughout the body. What happens when the body lacks enzymes? Lack of enzymes in the body may include lack of muscle coordination, brain degeneration, learning problems, loss of muscle tone, increased sensitivity to touch, feeding and swallowing difficulties, slurred speech, and an enlarged liver and spleen. What disease is caused by enzyme deficiency? Now, we have a disease called the Gaucher disease, which is a rare inherited metabolic disorder in which deficiency of the enzyme called glucocerebrosidase results in the accumulation of harmful quantities of certain fats, lipids, specifically the glycolipid glucocerebroside, throughout the body, especially within the bone marrow, the spleen, and the liver.
And the biological importance of hydrolysis. Now we are going to look on the reactions. Hydrolysis catalyze hydrolysis reactions. Hydrolysis disrupt the chemical bond by adding water. This is the acylation step. And also hydrolysis reactions can be used to cleave any type of bond, especially peptide bonds, to break down proteins and polypeptides in such reactions. Other hydrolysis reactions can be used to cleave phosphodiester bonds to degrade DNA molecules. Such reaction is performed by restriction enzymes, also known as restrictive endonucleases. Hydrolysis helps to induce degradative reactions in the body. For example, during digestion, lipase hydrolyzes lipids and proteases converts proteins to amino acids. Hydrolysis also cleaves large molecules into fragments used for synthesis, excretion of waste materials, or as sources of carbon for production of energy.
That is all we have for today. In summary, we spoke on the class of enzyme hydrolase, and we made you to understand that they perform as biochemical catalysts that use water to break chemical bonds. We also told you that under the enzyme commission number of classification, hydrolyses are classified as EC3, and they are further subclassified into 13 subclasses, ranging from the EC 3.1 that includes enzymes like esterases and lipases that cleave on ester bonds to the EC 3.13 that act on carbon sulfur bonds. Examples of hydrolysis were also mentioned to include all the digestive enzymes we know that are in the body. Further through this video, the function and importance of hydrolysis were mentioned. Generally, they are pivotal to the body since they digest large molecules into smaller fragments. Chemical reactions and structures of hydrolysis were mentioned, and several functions of hydrolysis were listed, but we generalized them to include that hydrolysis participates in a variety of biological procedures due to their diversification of action. Ladies and gentlemen, we have come to the end of this presentation, and I hope you have been able to enlighten you on this special class of enzyme known as hydrolysis. I would like to thank each and every member of this group for putting in effort to make this great assay a huge success. And to you out there watching this video, I say a very big thank you to you, and we hope to expect a positive feedback from you. That will be all for today. Thank you and enjoy your day.